Now, what is the role of tapetum? Tapetum is an important layer which provides nutrition, or it acts as nutritive tissue for the developing microsporangium. It also secretes some of the amino acids, some hormones, and some enzymes. In angiosperms, the tapetum, it is of two types: glandular or secretory tapetum, which secretes certain substances that help in dehiscence of anther, and the amoeboid tapetum, which helps in dissolution of the walls between microsporangium. Now, in when the anther is young, a group of compactly arranged homogeneous cells, that is the sporogenous tissue, it occupies the center of microsporangium. देखो हमने ये देखा कि ये था हमारे पास stamen with a filament and anther. Each anther has two anther lobes, और उन anther lobes में क्या था, उनके सेंटर में the circular microsporangium. Microsporangium was protected or surrounded by four layers: epidermis, endothecium, middle layers and tapetum. इसमें epidermis, endothecium और tapetum, they are single layer, they are made up of one layer of cells, and middle layers जो हैं ये 2 से 3 layers हैं cells की, and these layers they perform different functions. Now when the anther is young, the tissue inside each microsporangium, that is the sporogenous tissue, it occupies the center of the microsporangium. Now the cells of these sporogenous tissue, these cells of sporogenous tissue, they undergo meiosis, and this results in the formation of 4 haploid cells, and this is called microspore tetrad. Each cell in this sporogenous tissue, it is capable of undergoing meiosis and giving rise to a microspore tetrad. तो हर सेल को, स्पोरोजीनस टिश्यू के हर सेल को हम क्या बोलेंगे? दे आर कॉल्ड माइक्रोस्पोर मदर सेल. ठीक है, माइक्रोस्पोर मदर सेल. लेट अस सी हाउ दिस मियोसिस टेक्स प्लेस. ये हमारे पास है, क्या है? दिस इज द सेल ऑफ स्पोरोजीनस टिश्यू. नाउ इट अंडरगोज meiosis. First is the meiosis 1. Meiosis 1 results in the formation of 2 nuclei, and then क्या होगा? There will be meiosis 2. Meiosis 2 results in the formation of 4 nuclei, and these are the 4 haploid nuclei. This is followed by cytokinesis, that is the division of cytoplasm. तो ये क्या बन गया? ये क्या था? सेल माइक्रोस्पोर मदर सेल जो गया, which has undergone meiosis. It resulted in the formation of 4 haploid nuclei. और फिर क्या हुआ? साइटोकाइनेसिस, यानी अब हमारे पास क्या बन गए? 4 हैप्लॉइड सेल्स. और ये क्या है? दिस इज द माइक्रोस्पोर टेट्रड. दिस इज द फॉर्मेशन ऑफ माइक्रोस्पोर टेट्रड. और बीच में ये जो सेल है, ईच सेल इज कॉल्ड माइक्रोस्पोर. ऑल दीज 4 cells are microspores which are haploid in nature. Now, the process of formation of microspores from microspore mother cell, this process is called microsporogenesis. What is microsporogenesis? Formation of microspore tetrad from microspore mother cell by the process of meiosis is called microsporogenesis. अब ये microsporogenesis हो गई. इसके बाद क्या है? The microspores, as they are formed, they are arranged in cluster of 4 cells. This is how they are formed, and this is called microspore tetrad. As the anther matures, it dehydrates the microspores inside the microsporangium. They are formed and they are released with the dehiscence of anther. Let us see how. ये था हमारे पास स्ट्रक्चर. दिस इज द स्ट्रक्चर ऑफ एंथर, मैच्योर एंथर. इतने पोर्शन को, ये लेयर्स को एनलार्ज्ड व्यू दिखाया इसका, जिसमें आउटर मोस्ट इज एपिडर्मिस, देन एंडोथीकियम, फिर ये है हमारे पास मिडल लेयर. मिडल लेयर के बाद दिस इज द लेयर कॉल्ड टैपिटम. टैपिटम के अंदर है स्पोरोजीनस टिश्यू, और हर सेल स्पोरोजीनस टिश्यू का जो है दैट इज माइक्रोस्पोर मदर सेल. और उसके बाद जब ये फॉर्मेशन हो जाएगी, किस फॉर्म में होगा? इन द फॉर्म ऑफ दिस टेट्राड. और जब पूरी डेवलपमेंट कंप्लीट हो गई, इट विल ड्राई अप, dehydrate, and the dehiscence will take place. It will result in the liberation of the tetrad, that is microspore tetrads and the pollen grains. जो ये एक एक सेल, जो ये टेट्राड का हर सेल था, now it will develop into a pollen grain. This is how the dehiscence takes place and the pollen grains are formed. Now, pollen grains, they are generally spherical structures which are about 20 to 25 microns in diameter. Each pollen grain is surrounded by 2 layers. One is the outer hard layer, which is called exine, and this exine is made up of sporopollenin. Sporopollenin, it is the most resistant organic material. It is such highly resistant that it can withstand very high temperature, strong wind, strong acids and alkalis. यानी कि exine which is made up of sporopollenin, ये sporopollenin is resistant which can withstand very high temperature. यहाँ तक कि एसिड और अल्कलीज को भी ये जो है उनको भी रेजिस्टेंट है, यानी उनको भी बर्दाश्त कर लेता है. एंड no, there is no enzyme which can degrade sporopollenin. The pollen grain, it has got prominent apertures. जहाँ जहाँ पे exine या sporopollenin present नहीं है बीच बीच में, it is, exine is not a continuous layer. At places the sporopollenin is absent and these pores are called germ pores. ठीक है? And there is inner wall of pollen grain which is called intine. Intine, it is a thin layer which is made up of cellulose and pectin. Cellulose and the pectin. Now, यहाँ पे ये हमारे पास है exine, intine, and then there is, this is the cell membrane of the pollen grain.